Genetic secrets of almost 2,700 cancers unveiled by Landmark International project scientists have revealed the detailed genetic makeup of thousands of cancer samples, yielding new insights into the genes that drive the many and varied forms of the disease. The results, published in a landmark collection of research papers in the journal Nature, interpret the complete DNA sequences, or cancer genomes, of 2,658 cancer samples. This will further our understanding of the crucial driver mutations that underpin cancer development and offer potential as targets for treatments such as chemotherapy. It is the work of some 700 scientists around the world as part of an international project called the Pan-Cancer Analysis of Whole Genomes. The hallmark of a cancer cell is its unregulated growth. The mechanism that allows these cells to escape normal cellular growth regulation involves the introduction of mutations into the cancer cell's DNA. The collection of mutations present in a particular cancer genome is thus known as that cancer's mutation signature. Each advance in our capacity to accurately and completely sequence whole cancer genomes and to analyze the sequence data has enabled a more in-depth analysis of these mutation signatures. Each step forward has revealed further diversity in the mutation processes that underlie the development and progression of cancer. Diverse mutations. It is 7 years since the previous landmark advance in this field. Back in 2013, researchers reported on the genetic makeup of 7,042 cancers of 30 different types and identified 20 distinct mutational signatures. Today's reports involve fewer cancers, but an increase in the number of cancer types to 38. But this latest advance is not really about numbers. The real step forward is in our understanding of the diversity of DNA mutations and mutation signatures within cancer genomes. This is primarily the result of improved methods for analyzing the DNA sequence data compared with the state-of-the-art in 2013. As a result, important DNA sequence alterations that could not be detected in previous work have now been described. Each contributes important new details about each cancer genome. Until recently, cancer DNA mutation analyses had been focused on small alterations in coding regions of DNA, the roughly 1% of DNA that is responsible for making proteins. The new analyses reported today have identified non-coding driver mutations, some of them large structural mutations that can be as big as entire chromosomes. These new analytical capabilities have enabled the identification of 97 mutation signatures, 5 times more than previously known. The improved detail boosts our understanding of the diversity of cancer genomes. It also provides important new information about the order in which these mutations accumulate during cancer development. However, there is good evidence to suggest that more work is still required to characterize the full spectrum of cancer DNA mutations. It is anticipated that all cancers will have at least one, and perhaps as many as 5, driver DNA mutations. Despite the extensive array of analytical approaches described in these new reports, the researchers were still unable to identify any driver mutations in 5% of the cancers in their study. The research has also shown that similar mutation signatures are present in cancers that arise in different tissues. This has implications for cancer treatment. For example, a drug successfully used to treat a breast cancer may be as effective for treating a pancreatic cancer if the two cancers share the same mutation signature. These data will greatly advance our ability to identify cancers with the same or similar origins via their mutation signature. It has enormous implications for diversifying the current suite of drugs available for gene-targeted cancer treatment. But, perhaps more significantly, it also offers the opportunity to expand our strategies for preventing cancer before it starts. Why the causes of cancer are more than just random bad luck. What causes cancer? This deceptively simple question has a devilishly complex answer. So when US researchers proposed a relatively simple mathematical formula to explain a long-standing conundrum in cancer earlier this year, it was bound to get a lot of attention. The study published in the journal Science suggested a correlation between the variation in cancer occurrence between different tissues and the number of stem cell divisions in each tissue. In other words, it said the tissues most vulnerable to cancer are those with the greatest number of stem cell divisions. Most of the reporting about the research ran with the line that cancer is all down to bad luck, implying that developing the disease is out of our hands and that preventative efforts might be useless. But is that really the case? Much of the misunderstanding seems to have arisen from the author's statement that a third of the variation in cancer risk among tissues is attributable to environmental or inherited factors, with the majority due to random mutations during DNA replication in normal cells. This statement about relative risk was overblown into blanket conclusions about the underlying causes of cancer. The wonder of replication cancer emerges when one of the cells that make up your tissues and organs grows and divides without control, losing its specialized function and invading other tissue. This happens when normal control of cell growth and division is compromised through changes or mutations in your genome, the chemical instruction book for life. Mutations lie at the heart of cancer biology. The genome is made from a chemical alphabet of just 4 letters, A, T, G, and C, written into DNA. It works like a kind of computer software for our cells, with strict instructions for growth and function. Each of the 100 trillion cells in your body contains roughly 6 billion letters, called nucleotides, of this code, condensed into a thin strand of DNA about 2 meters long. To put this into perspective, if you stretched out all the DNA in a human body, it would reach around the moon and back several times. Every time a cell divides, the genome must be copied accurately and quickly. This synthesis of new DNA is called replication, and the numbers behind it are staggering. UK researcher John Diffley has calculated that you will have synthesized the equivalent of a light year of DNA, 10 trillion kilometers, by the time you're 50. Words simply cannot do this amazing process justice, but this short video by award-winning animator Drew Barry will blow your mind. DNA replication has evolved to be incredibly efficient and reliable, but random mistakes, mutations, occasionally happen. Still, they occur at a rate of less than once per genome per cell division, thanks to some impressive molecular proofreading machines, which constantly survey the newly copied DNA and correct errors. But with so many cells dividing so often, DNA replication still represents a major source of mutations. And every cell division increases the chance of accumulating mutations in important genes, increasing the likelihood of cancer. Other sources of mutations can take many forms and can emerge in a number of ways, not just through replication errors. We inherit between 50 and 100 mutations from our parents at birth, for instance, and any new or de novo mutations act on this inherited genetic background. Even normal cellular metabolism damages DNA through the production of reactive oxygen. And, in a sinister twist, many of the inherited mutations that predispose people to cancer hit genes that control the DNA proofreading and repair systems, such as the breast cancer genes BRCA1 and BRCA2. This has the effect of amplifying the rate of new mutations. The other major causes of DNA mutation are lifestyle or environmental factors. We are exposed to a range of these in our everyday lives, such as UV radiation from sunshine and chemicals including asbestos or from smoking cigarettes. Lifestyle factors including diet and alcohol consumption may also contribute. Some viruses and bacteria are known to cause DNA damage leading to cancer. They include the human papillomavirus, HPV, for cervical cancer, and H. pylori for gastric cancer. Although these different agents leave unique chemical signatures in the DNA, they are still essentially random events. Random mutations are, in fact, the raw material driving evolution. And the processes of mutation and evolution are accelerated in cancer. Indeed, we are only now starting to understand the importance of evolution in driving cancer emergence and spread, as well as its resistance to therapy. Minimizing risk. Where does this leave the idea that cancer is all down to bad luck? Is modifying your lifestyle to minimize exposure to risk factors futile? As usual, reality lies somewhere in the middle of competing narratives. Life is a kind of genetic gamble. We have to play the cards dealt us, but we can stack the odds in either direction by altering our exposure to environmental and lifestyle factors. Suggesting cancer is all down to bad luck dilutes the important message that risk can be modified by behavior. The Cancer Lexicon is littered with notions of guilt and blame. Death is often framed as losing the battle with cancer, for instance. And patients and their families are bombarded by gurus profiteering from various diet and lifestyle interventions. Their implicit messages can often leave people feeling that their cancer is all their own fault and wondering if there was something they could have done differently. The fact remains that, in many cases, there isn't. Cancer signatures offer hope for treatment, and prevention research published in the journal Nature overnight describes the mutations that make cancer cells grow faster than ordinary cells. These mutational signatures don't just open up avenues for better cancer treatment, they also provide information that could help us refine cancer prevention efforts. The hallmark of a cancer cell is its unregulated growth. The mechanism that allows these cells to escape normal cellular growth regulation involves the introduction of mutations into the DNA of the cancer cell, which is known as the cancer genome. This mechanism of genetic change is called a somatic mutation. Somatic mutations are genetic mutations that are not inherited but arise in cells during life. In some cancers, a substantial proportion of somatic mutations are known to be caused by exposures, such as tobacco smoking and lung cancer. But the process that causes these somatic mutations in most cancers is not well understood. Technology enabled as technology has developed over the last many decades, we have gradually started to understand the nature and diversity of somatic mutations that arise during a lifetime, including those that are significant enough to cause and promote cancer development. Technical advances in the preparation and visualization of chromosomes during the 1950s and 1960s provided evidence that most cancers were associated with changes in chromosomes, tightly curled strips of DNA within the nucleus of cells. A noteworthy example was the identification of the Philadelphia chromosome, a defective chromosome 22, named after the city in which it was discovered, associated with chronic myeloid leukemia. Over the last 50 years, researchers have gained a better understanding of the Philadelphia chromosome, and chronic myeloid leukemia has been successfully treated with imatinib mesylate, commonly known as Gleevec. This drug targets the specific genetic abnormality found in chronic myeloid leukemia. The success of this kind of targeted therapy in treating chronic myeloid leukemia stimulated the research community and pharmaceutical industry to pursue similar opportunities to advance cancer treatment and founded the current era of personalized medicine. New working models of cancer development supported by limited but increasing amounts of information about somatic mutations have developed extensively during the last few decades. In 1980, Bert Vogelstein proposed that cancer was caused by sequential mutations in specific genes. This idea was supported by genetic information he had collected from colorectal cancers and applied an insightful evolutionary perspective to cancer development that remains robust today. He suggested that somatic mutations gave some cells a growth advantage over cells without the mutation. And when these types of mutations accumulated in a cell, it was eventually able to escape normal cellular regulation, the hallmark of cancer cells. Another enormous advance in genetic technology has now facilitated the research published in Nature to provide a significant and extensive analysis of the somatic mutations that arise in cancer cells. This new genetic technology is known as massively parallel sequencing, and it can affordably sequence whole cancer genomes in a matter of days. It has revolutionized genetic analyses that had previously relied upon much more laborious and expensive technology, such as Sanger sequencing, developed by Frederick Sanger in the 1970s. This technology has also spawned new approaches to data analysis and, in the new report, the researchers have developed methods to describe mutational signatures, groups of somatic mutations, that are found together in cancer cells. What we've learned: these advances enabled the analysis of 7,042 cancers, including 30 different cancer types, and the identification of 4,938,362 somatic mutations. They revealed more than 20 distinct mutational signatures, illustrating substantially more diversity in the somatic mutation process underlying the development and progression of cancer than has ever been described before. Many aspects of the new findings are fascinating. Some cancer types carry very few somatic mutations, some childhood cancers, for instance, while others have an extensive collection of somatic mutations, such as those related to prolonged exposure to substances such as tobacco smoke. This can be attributed to a lifetime's collection of somatic mutations or the rate of somatic mutation accumulation. Most cancer genomes that were sequenced contained more than one mutational signature. And some of these were found in the majority of cancer genomes, while others were much more specific to the type of cancer. The authors suggest that each mutational signature is the imprint left on the cancer genome by the somatic mutational process, and that it is likely that more mutational signatures will be identified with further improvements in genetic